Okay, last talk before you can go to the beer session. I hope you still have some energy and attention span left to listen to another talk. I must admit I'm completely done and saturated. Right, all the virtual conferences that we had, maybe not good for our training and concentration, but I hope I can entertain you a little bit. There's a talk that should have, for almost everybody, some of interest in there. There is NMR in there, there's crystallography in there, cross-linking mass spectrometry. The only thing that is missing, Shores, is cryo-EM. Sorry. Okay, so in this talk I will tell you about our discovery of a novel function, a novel mechanism of histone chaperones. And this morning in the beautiful talk of Ben Schuler, you were already introduced to histone chaperones, and he was describing histone chaperones specific for the linker histone H1, but most of the histone chaperones in the cell target the core histones that are inside at the heart of the nucleosome. And in my group we're interested in chromatin, and specifically in the proteins that bind to chromatin, because they really bring the whole thing alive. They remodel the structure of chromatin, recruit all the enzymatic activities that are needed to regulate important things like gene transcription and DNA repair. And in my group we use an NMR-driven integrative structure biology approach to understand how these proteins operate on the nucleosome. So in this talk I focus on DNA repair. Every day, every cell in our body is faced with about 10 double-strand breaks that need to be repaired. They are very, very dangerous to the cell, to the genomic integrity. So the cell has, of course, a mechanism to repair that. But the presence of chromatin does create an additional challenge for DNA repair. First, some of these nucleosomes will have to be removed in order to make room for the DNA repair complex. And then after the DNA break has been repaired, these nucleosomes will have to be put back. The chromatin will need to be restored in order to maintain the proper packaging of the DNA and to maintain the epigenetic information. Now, this is why the DNA repair process also involves dedicated protein machinery to manipulate these nucleosomes and the histone proteins. And one important class of these proteins are so-called histone chaperones. And these histone proteins are actually weird proteins. When they were first discovered, they were not even considered to be true proteins because their physiochemical properties are just so different. And the funny thing is that as long as they are in the nucleosome, they're happy. But if you put them alone, they basically are very unhappy and will rapidly aggregate and precipitate on the DNA. So this is where these histone chaperones come in to keep them happy. By binding to the histones, they keep them soluble and okay. These histone chaperones also guide the assembly of the nucleosome. And the current paradigm is that this happens in two steps. That there is first a dedicated chaperone to deposit the H3-H4 unit onto the DNA, and then in the second step, a different chaperone may deposit H2A, H2B on this complex to form the ultimate nucleosome. So if we now go back to the DNA repair complex, specifically the one of the nanomolecules N joining, NHEJ, then it was found that one of the proteins in this complex, this purple protein, APLF, actually has also histone chaperone activity, and that this histone chaperone activity depends on the presence of this so-called acidic domain, or AD. Now, the intriguing thing is that the paper that described this showed that, or suggested that this histone chaperone, on one hand, is specific for H3, H4, but on the other hand, it also suggested that this protein is involved in the deposition of H2A variants that are specific to DNA damage. So this is actually in conflict with the canonical paradigm of histone chaperone function. So we wanted to understand this better, and this has been the PhD project of Ivan Korbeski in my team. And what he showed to begin with is that this APLF acidic domain, APLF-AD by itself, is also a histone chaperone. If you add histones to DNA, you cannot see any soluble DNA here anymore, but if you add in the APLF-AD, you can rescue the DNA from precipitation. He also then showed that this histone chaperone is actually neither specific for H2A and H2B, H2A, H2B, or H3H4. It binds both of them with very similar affinities and enthalpies. So then we went on with the NMR study, and as was shown earlier, if you see an NMR spectrum where the chemical shifts are in a very narrow range, that is a sign that you have an intrinsically disordered protein. So this APLF acidic domain is an IDR. That's not surprising. It has a very low complexity sequence. And then if you have these assignments, we can add in the H2A, H2B histone complex. And what happens then is mostly a disappearance of NMR signals. And if you plot that, you can determine the binding site for this histone complex. Now, if you add in H3H4 in a separate experiment, you get also peak intensity decreases, but in a different pattern, and that reveals that there's actually a distinct binding site for H2A, H2B, and a distinct binding site for H3H4. And that, of course, suggests that this protein may actually be able to bind both of them simultaneously. So he also tested that using analytical gel filtration. And if you just take these histones alone and you mix them, what you get is two peaks, one corresponding to this H3H4 unit and one to the H2A, H2B unit. But if you then add in APLF, acidic domain, what you see is that you get one large complex. And further experiments using native mass spectrometry and SAX showed that the mass of this complex is 123 kilodalton and is roughly globular with a maximum dimension of about 10-11 angstroms. So at this point, Ivan really started a massive effort to try and crystallize this protein. And in the end, with help of Hugo at Tisha Sixma lab at the NKI, he succeeded in that when using tailless histones and a truncated fragment of this acidic domain. And Xiaohu basically solved the structure and got it refined to about 2.3 angstrom. And when we saw this, it was really quite spectacular because it showed how APLF tethers together the H3H4 unit and the H2A, H2B unit in a completely novel manner. And what you can see here that there is two copies of APLF in the complex, and also two copies of H3H4, one here, one there, and there's also two copies of H2A, H2B, and here and there. So what you can also maybe appreciate that this complex is not only held together by interactions from APLF to the histones, but that there are also interactions between the histones themselves. And in fact, if you compare the arrangement, the conformation of the histones is exactly the same as in the nucleosome with 0.5 RMSD on the backbone. APLF is sitting on the DNA binding surface of the histones, so it provides a steric obstacle for DNA binding, but because it's an acidic domain, it's also very negatively charged, so it also basically forms an electrostatic block against DNA binding. Now, if we zoom in a little bit more into the interaction, wow, this looks really weird. Oh no, now it's fine. I had a weird glimpse there. But what we can see here, this is the crystallized part of the acidic domain, that there is actually a very important role of aromatic residues in this interaction. There's a tyrosine over here, 462, phenylalanine over here, 468, that are buried in a hydrophobic pocket on histone H3, and there's a tryptophan over here, 485, that is on a shallow hydrophobic surface on H2B. And these hydrophobic interactions are then complemented by really an extensive network of hydrogen bonds and salt bridges. Now these aromatic residues, they are critical for histone binding, so if you mutate them and check the affinity and binding enthalpy, then we see that if we mutate the anchors in the histone H3-H4 interface, so a double anchor mutant DA34, we lose binding affinity, we also lose binding enthalpy, suggesting that there's a defect in the assembly of the histone octamer. Similarly, it's sort of also still true for when you mutate the anchor in the histone H2A H2B interface, and if you combine the two together, you get, of course, the biggest defect in binding affinity and binding enthalpy. And this effect was actually even stronger in cells, and this experiment was done by Wouter Wiegand in the lab of Heiko van Auticum at the LUMC. And there we see that if we have cells that express EGFP APLF, that you can pull down histone H2B and histone H4 when you use the wild type protein. If we delete the acidic domain, no histones are pulled down. And even if we just do this quadruple anchor mutation, all binding of the histones is lost. And even more beautiful in this data, if you analyze it carefully, is that for these double anchor mutants that only target the anchors that are needed to bind histone 3,4 or AB, both histone H2B binding is reduced and histone H4 binding is reduced in both cases. And this really strongly suggests that also in cells, this chaperone binds to histone octamers rather than distinct AB or 3,4 units. So this is already great, but still left us with some questions, in particular because the crystal structure used only a truncated fragment of APLF-AD. So we were wondering what the C-terminal region is doing, as it is also highly conserved. So we went back to NMR, and this time we took APLF and titrated in a mixture of histone H2A, H2B and histone H3, H4, so an octamer mix. And even when you add in just a little bit of this material, the peak intensity drops dramatically as you form this 120 kilodalton complex. And the surprising thing here is that it's not only true for the histone binding region, but also for the C-terminal region, the C-tail. So that suggests that this is also close to the octamer surface. And moreover, we did relaxation dispersion experiments, and these show that residues in the C-tail also experience large changes in their chemical environment upon binding, and that they in fact feel the same binding kinetics as residues in the histone binding regions. So it's a concerted binding event. And then Domenico Vaschi from Albert Hecht's group at the UU, he could find some reproducible cross-links from the C-tail to histone H2B. So all this data together clearly shows that the C-tail is also involved in binding the histone octamer. So using this data, we then extended the crystal structure and used Haddock to model a structure of the full APLF acidic domain bound to the histone octamer. And what this shows is 20 different possible solutions that satisfy our experimental data. What this shows is that APLF basically envelopes, wraps the complete histone octamer, except this region here at the top that is close to the symmetry axis of the complex, which is the H3-H4 diet region. And this is very intriguing because it basically suggests to us that APLF can block DNA binding here, everywhere, except here. So it essentially could guide DNA binding to happen only at this region that also is the central region of the nucleosome. So to test this, we got really great help from Bruna Eckhart and Francesca Mattiroli from the Utrecht Institute. And Bruna devised an assay to test whether the APLF-AD octamer complex could bind DNA. And indeed it can. If you use a short piece of DNA, 50 base pairs, you can get this super shift formed by a ternary complex of APLF, octamer, and DNA. And the formation of the supershifted band only happens if you have the wild type protein. If you have the quadruple anchor, it doesn't occur. Now, that's also cool, but even more cool is that if you use a longer piece of DNA, that is the length of the DNA that's inside the nucleosome, APLF is actually excluded from the shifted band, and we see only the octamer and the DNA in the shifted band. So here we used fluorescently labeled proteins in order to identify each of the three components in each band upon native gel electrophoresis. So this very strongly suggests that what happened here is that nucleosomes are formed. Now, to really test this, Bruna did the test to assess nucleosome formation, and that is to try and chop them up. That's called micrococcal nuclease digestion. So this enzyme digests any free DNA, and if you have a true nucleosome, what should happen is that fragments of about 120 to 150 base pairs of DNA are protected from digestion. So if you have a true nucleosome, you do the assay, you get a lot of fragments in this range of 120 to 150 base pairs. Now, when she did this with APLF and histone mixtures, she could see that indeed we get more of these true nucleosome fragments compared to control, indicating that APLF-AD can indeed stimulate nucleosome formation. So with all this data, we then can put it together and propose a new mechanism, a novel chaperone mechanism for APLF-AD. So in contrast to the canonical mechanism where we have a stepwise assembly of the nucleosome, first H3, H4, and then H2A, H2B, APLF can single-handedly assemble the histone octamer, direct the DNA to bind at the H3, H4 diet axis. The DNA will have a higher affinity for the histones than APLF, so it can basically peel off the APLF, resulting ultimately in the deposition of this octamer onto the DNA and the formation of a nucleosome. So what is really the interesting point here is that in the canonical mechanism, this octamer assembly only happens coincident with nucleosome assembly, and in the APLF mechanism, the histone octamer can be assembled off the DNA and is completely decoupled from nucleosome assembly. So to conclude, we gathered lots of structural data, biochemical data, and cellular data to integrate it into a novel chaperone mechanism, that is the chaperoning of the entire histone octamer. And APLF can do that because within this acidic domain, there are a few aromatic residues spaced just right to bind both H2A, H2B, and H3, H4 and tether them together to stabilize this histone octamer. And by wrapping around this histone octamer, it can prevent non-native contacts, guide the DNA approach, so to promote nucleosome assembly by depositing this histone octamer onto the DNA. Now, we looked at other histone chaperones, either that are known in structurally in the PDB. They all are incompatible with this binding mode. There are also other chaperones that have acidic sequences. There it's a little bit harder to tell whether they can or cannot or could not use this mechanism, but neither of them has a very clear alignment to the sequence of APLF-AD. So it could be that other chaperones can also do this, and that it is a more general widespread mechanism to maintain the histone octamer during chromatin disruptions. And within the DNA repair complex, we think that APLF can capture effective histones, and because there's two APLF molecules as it binds to the DNA end binding protein Q, it can also assemble these then into a histone octamer, and we propose that it will then promote chromatin restoration after repair by stimulating nucleosome formation. So with that, I'd like to thank especially Ivan, who did really a marvelous job. He's now a postdoc in Basel. I'd like to thank all my colleagues in the NMR group in Utrecht, and especially also the great collaboration. Really did an amazing job, and it was a lot of fun to work with all of them. And I'd like to thank INEX Discovery and Instruct for their support, and you for your attention.